Welcome back to the playlist on the respiratory chain. In this video, we're going to go over the mechanism and physiology of one of the enzymes that's in the respiratory chain of the mitochondria called Cytochrome C Ubiquinol Oxidoreductase. You might also hear this enzyme referred to as complex 3, although you do need to know both of the names. And this particular enzyme mechanistically is going to go through something called a Q cycle. Okay, the Q cycle is very important to understand because what we're going to get is a division of electrons, meaning the electrons from this molecule right here, which is called coenzyme Q in the reduced state, they're going to split. One's going to go one direction and the other's going to go another direction, and we'll see how that works in a few minutes. Now, the coenzyme Q, or the ubiquinol, that is essentially used by this enzyme, keep in mind that that ubiquinol essentially came from complex 1, which is called NADH dehydrogenase, and then other enzymes like succinate dehydrogenase also produce this coenzyme Q in the reduced state, okay? And there's some other ones that do as well, like electron transferring flavoprotein ubiquinol oxidoreductase. They all lead to this increase in coenzyme Q in the reduced state, and this guy sort of moves in the membrane and meets up with cytochrome C ubiquinol oxidoreductase complex 3, and it's able to use it. Now, one complete cycle of this enzyme is going to require 2 coenzyme Qs in the reduced state and 2 cytochrome Cs in the oxidized state. And in the next video, we'll look at a lot of the function of cytochrome C and how it's made and so forth. Essentially what cytochrome C is it's a heme-containing protein. So cytochrome C, let me actually come over here and do this. So cytochrome C, just to give you some background information, this is a non-enzyme. Okay, it's a protein, but it's non-enzymatic. Okay, it doesn't have any catalytic properties. Okay, what it does is it has this prosthetic group called heme C. Okay, and in the next video, if you want more detail on this, you can certainly go watch that. We go over the synthesis of cytochrome C and then also physiology of heme C. What heme C does is it picks up electrons. So for example, you could have heme C with iron in the 3+ state, and if it picks up an electron, right, it becomes heme C with iron in the 2+ state, and it can interconvert with that if it loses an electron and so forth. Okay, so basically there's 2 forms of cytochrome C. One where the heme iron is in the 3+ state, another where the heme iron is in the 2+ state. The cytochrome C form where it's in the 2+ state, you basically call this ferrocytochrome C. If it's in the 3+ state, you refer to it as ferricytochrome C. And the origin of that is that when the heme is in the 2+ state, that's ferrous iron. The O goes with the O. If you're in the 3+ state, that's the ferric state of iron. I goes with I. So ferrocytochrome C is the reduced form. Ferricytochrome C is the oxidized form. Okay, so basically in this mechanism, we're going to start out with cytochrome C in the ferricytochrome C form. It's going to be in the oxidized state, and it's basically going to pick up the electrons from coenzyme Q in something referred to as the Q cycle. And down here, we have some different kinds of inhibitors of this enzyme, which are basically pretty much lethal for the most part. And we'll go over that after we do the mechanism. But to understand how these guys work, you at least have to see the mechanism of this enzyme. So let's do that now. Okay, so some important things to realize about this enzyme. Number one, we have this critical histidine residue in the active site. So there's a histidine residue right here. Usually in this type of enzyme, it's approximately amino acid number 161. So I will consistently try to say this particular histidine as 161. So his 161. And this his 161 has this Risk iron sulfur center. So that's what this is. These 2 irons and these 2 sulfides that are electrostatically interacting with the nitrogen here. This is called the Risk iron-sulfur cluster or iron-sulfur center. And a lot of the redox reactions are going to take place using one of the ions of this Riesk iron-sulfur center. Okay. There's also a critical glutamate residue in the active site that begins the mechanism in the deprotonated state. Both the histidine and the glutamate are going to act as Bronsted-Lowry acids and bases. Okay. They're going to be proton exchangers. The Riesk iron-sulfur center is going to be for the electron transfers. Okay. So let's actually look to see how it works right now. Okay, so basically in the first mechanistic step, which I'll do in green, okay, glutamate is going to deprotonate this molecule right here, which is called ubiquinol. Okay, so this molecule right here, this is totally reduced ubiquinol. Sometimes you'll hear it referred to as coenzyme Q in the reduced state, but in any case, it's ubiquinol. And what ends up happening is this glutamate residue deprotonates, and you end up with a lone pair on that oxygen. Also, what's going to happen is this histidine is going to deprotonate this oxygen, this hydroxyl group of the ubiquinol, but instead of just going up as a lone pair on the oxygen, you're going to get a homolytic bond cleavage. So let me do this in a different color. So one of these electrons ends up as a radical electron on this oxygen of the ubiquinol ring. The other one of those electrons goes and reduces one of the iron cations of the Riesk iron-sulfur center. And so notice how both of these irons right here are in the 3+ state. So you would imagine that the Riesk iron-sulfur center would have one of them reduced because one of those electrons came off of the ubiquinol and went to the Riesk iron-sulfur center. So that's what you see here. One of them is in the 3+ state, but this guy right here is in the 2+ state. Okay? So hopefully that makes a little bit of sense. Now, there's another pathway that electrons can go, and they can go through a series of hemes. So heme 401 in the case of this enzyme, this is called heme low potential, often abbreviated H sub L. Heme 402 is going to be heme sub H, H sub H, high potential heme. Okay? And there's also a totally oxidized ubiquinone in the active site. So this guy right here, this is ubiquinone. Okay, the -one signifies that the quinoid molecule is in the oxidized state, whereas when you say quinol, this signifies that this is in the totally reduced state. So there's a ubiquinone in the active site. And so what's going to happen is this lone pair that's on this alkoxide here is going to collapse back in. And so you're going to get a double bond rearrangement, and when you kick out here to form the carbonyl, this electron right here first kind of jumps over to the heme low potential, then it jumps to the heme high potential, and then essentially what's going to happen is it's going to nucleophilically attack this oxygen right there. And that's going to force a double bond rearrangement, so this comes in right there, that comes in right there, and you end up generating an oxyanion intermediate. Okay, so this intermediate right here, this is called the semiquinone. This is the semiquinone intermediate of CoQ. Okay, it's a semiquinone. And the reason it's called the semiquinone is because it's a radical. So whenever you have a semiquinone radical, you notice that one of these oxygens right here has a radical electron. Of course, there are 4 other electrons, 2 lone pairs on it. Overall, the oxygen is neutral, but it turns out it can actually stabilize that radical electron. Okay, so having said that, now what's going to happen is we're going to attempt to regenerate the resting state of the Bronsted-Lowry acid-base residues in the active site. So water molecules are going to come in. One of them is going to deprotonate this glutamic acid, regenerating the deprotonated glutamate, and the other water molecule is going to deprotonate this critical histidine residue, regenerating deprotonated histidine 161. Okay? Now keep in mind, what ends up happening at this point is this iron is in the 2+ state as part of the Riesk iron-sulfur center. But in this step, here's the very critical part. This heme C right here, so this moiety right here, this is called heme C. This is cytochrome C. Let me actually write out the whole name just to illustrate the point. This right here is cytochrome C, and notice that the iron starts out in the 3+ state. Okay? So this literally is cytochrome C. Now cytochrome C will literally come and attach itself to this enzyme non-covalently. And one of these electrons from this ion of the Risk iron sulfur center, okay, it's going to be donated ultimately onto this iron in the 3+ state. And of course you would expect that if you have iron in the 3+ state and it receives an electron, now your heme C, this is also heme C, right? Your heme C is now going to be in the 2+ state. So now cytochrome C got reduced. So let me do this in a bold color. This right here, this is cytochrome C in the oxidized state. Okay, this over here, this is cytochrome C in the reduced state. Okay, so that's really critical to understand. And then this cytochrome C in the reduced state, just to give you some foreshadowing of the next few videos, this guy is going to go and react with cytochrome C oxidase, which you may also hear referred to as complex 4. This is complex 4 of the mitochondria, and it will be reoxidized back to cytochrome C in the oxidized state, and it can then go and pick up another electron from this enzyme. Okay, so cytochrome C comes by, picks up the electron from the Risk iron sulfur center, and then dissociates and goes to cytochrome C oxidase. And so you sort of have this going back and forth of cytochrome C between cytochrome C ubiquinol oxidoreductase and cytochrome C oxidase. So hopefully that makes sense to you. Okay. Now, if the iron of the Risk iron sulfur center donates an electron, it gets reoxidized back into the 3+ oxidation state. And in the process of doing this mechanistic step, you regenerate glutamate number 1 in the deprotonated state. You regenerate this Schiff base nitrogen also in the deprotonated state. But here's one key thing, is that you end up generating 2 hydroniums. Okay, so you end up generating 2 hydroniums. So these are H3O pluses right there. And what happens to these protons or these hydroniums? Well, these protons or these hydroniums, they end up getting pumped. These guys end up getting pumped into the inner membrane space of the mitochondria. And part of the reason that you need those protons pumped is because you need that concentration and pH and all the electric gradient to power ATP synthase, because it needs a lot of mechanical energy. And we'll talk more about ATP synthase in future videos. But the idea is that it needs mechanical energy, and the gradient you create by pumping protons is what does that. Now, one thing I just want to have you bear in mind is, you could effectively think of 2 hydroniums as 2 H+, although they exist in the hydrated form as H3O+. I want to think a little bit about where those protons ultimately came from. And the answer to that question lies in the fact that they both initially came from ubiquinol. The first ubiquinol that came into the active site, of course, had 2 protons on it. So those 2 hydroniums, and you can follow the mechanism if you want, but I think you'll see that both of those protons ultimately came from ubiquinol. Okay, and now they're pumped into the inner membrane space as hydronium. Okay, so at this point, what's going to happen is now another subunit of this enzyme, cytochrome C ubiquinol oxidoreductase, is going to allow 2 protons actually into the enzyme. And I'm actually going to circle those in a different color just so you can kind of distinguish between the two. Let me do it in orange. So this is an acid hydronium that's allowed into the enzyme, and so is this one right here. Okay, now here's what I want to show you. I want to show you this really good picture that let me zoom out a little bit. I found this picture essentially on Wikipedia, so just to give them credit for this. Okay, so notice here you have this Q sub 0 subunit and this Q sub I subunit. Okay, the Q sub 0 subunit, that's essentially what is picking up the protons ultimately from the ubiquinol. Okay, so notice how ubiquinol is coming into this particular part of the enzyme. And so this is initially what does the splitting of electrons, because notice electrons go this direction towards the iron sulfur center and then ultimately to cytochrome C, and some of them go this direction towards the low potential and high potential hemes. But the Q sub I subunit in our mechanism right now is about to pick up some protons. Okay, in fact, it's going to pick up 2 protons, as we'll see. 4 protons are essentially going to be pumped out eventually, but 2 protons are now going to come in. And they're going to come in as hydronium. And essentially the net effect is these protons are going to end up on a totally oxidized ubiquinone that we're going to allow into the active site from a previous cycle. Okay? So now to do this mechanistic step, number 1, we're going to allow another ubiquinol into the active site. So this is ubiquinol right here, totally reduced coenzyme Q. And a very similar thing is going to happen as to what happened in the last part of the mechanism. So number 1, we're going to get glutamate to deprotonate ubiquinol right here, and that's going to generate our oxyanion there. And then what's going to happen is this Schiff base nitrogen on the histidine is going to deprotonate this oxygen, and just like in the last case, I'll do this in yellow, you're going to get a homolytic bond cleavage. One of these electrons is going to end up actually on that oxygen. The other is going to reduce the Riesk iron sulfur center iron into the 2+ state. And eventually what we'll be able to do is pick up that electron using cytochrome C, and we'll see that in just a few minutes. Okay, but now here's something important to realize. What we have now in the active site of this enzyme is we have essentially two semiquinone radical CoQs. So 2 semiquinone radical CoQs, and it turns out that one of them is going to collapse. One of them is going to collapse, and what's essentially going to happen is this is going to form a carbonyl right there, so carbonyl formation, and then you're going to get double bond rearrangement. So this rearranges to right there. This rearranges to right here to form another carbonyl, and in the process, this electron that's right here is a lone electron on that oxygen. When carbonyl formation occurs, this electron is going to kick onto the heme low potential, heme 401, and then onto heme 402, which is the high potential heme, and then that's going to go onto here, onto this particular oxygen atom of the semiquinone radical. And then what's going to happen essentially is you're going to get proton transfers. So initially what will happen is this oxygen will then deprotonate this hydronium. The resulting hydronium bond breaks, which deprotonates this lysine in the active site. Now this particular oxyanion on the quinonoid ring is going to deprotonate this hydronium, but for now the water effectively will not deprotonate this histidine right here. It's going to wait and do that later. Okay? But in the process, notice what we generate. We generate this guy, which is ubiquinol. And we also generate a ubiquinone. And remember, ubiquinone is whenever it's in the oxidized state. So this is CoQ in the oxidized state. Ubiquinol is when you have CoQ in the reduced state. And that's pretty handy because what did we have to use as the initial substrate for this reaction? Well, you go back and you see, hey, I had to use a ubiquinol to start this reaction scheme. So it's pretty handy that this enzyme makes ubiquinol in the middle of its cycle. So it's not just NADH dehydrogenase, it's not just succinate dehydrogenase. No, this enzyme also makes ubiquinol in the middle of its Q cycle. So we end up generating that, and that can be used in the next cycle of cytochrome C ubiquinol oxidoreductase. But we're not done with this mechanism quite yet. So what ends up having to happen is you have to regenerate the resting state of this enzyme. So keep in mind, number 1, we have this histidine 161 in the protonated state. And we have to get that back to the deprotonated state. Also, we have this glutamic acid in the active site, and we have to get that back to the deprotonated state. Okay, so now what has to happen is you have to regenerate the resting state of the histidine residue, histidine 161. You have to regenerate the resting state of glutamate in the active site, And you have to get rid of that electron that's in the Risk iron sulfur center. So the way you're going to do that is, number one, you're going to use Cytochrome C. So I'll do that step. Let me do that step in yellow just to illustrate that it's super important. So what has to happen is an electron has to come from the Risk iron sulfur center, and it's going to reduce the iron of the heme C of Cytochrome C. And so what that generates is this. It's Cytochrome C in the reduced state, in the 2+ oxidation state. Okay. Also, what's going to happen, I'll go back to using green for my mechanistic steps, is there's a water that's going to deprotonate another water, which is going to deprotonate the glutamic acid in the active site. And that's going to generate a hydronium, and then a water will then deprotonate this histidine 161, regenerating deprotonated histidine and another hydronium. So far, if we're keeping track of our hydroniums, we have 3 of them. So now we have deprotonated histidine 161. We have successfully regenerated the Risk iron sulfur center to its original oxidized form. We have deprotonated glutamate, and now we have these 3 hydroniums that we have to deal with. Okay? And of course, the Cytochrome C in the reduced state goes and dissociates and goes over to Cytochrome C oxidase, which is going to be complex number 4. And it's going to give up that electron, each one at a time, to Cytochrome C oxidase, which will ultimately reduce molecular oxygen to 2 waters through 4 cycles of that. Okay? So now we have these 3 hydroniums. We have a deprotonated lysine, which we have to regenerate. And so what's essentially going to happen here is lysine is going to attack one of these protons on the hydronium. And so the net effect is getting lysine back to its protonated state, the way it started the mechanism, and you also get 2 hydroniums. So here's the key. What happens to these 2 hydroniums? Any idea? Well, we know that Cytochrome C Ubiquinol Oxidoreductase pumps 4 protons, and we already saw that at the end of mechanistic step 3, what ended up happening was you generated 2 hydroniums, and those got pumped into the inner membrane space. So just like in that case, these protons, these will get pumped out into the inner membrane space, thus fueling the proton gradient that ultimately will power the mechanical energy needs of ATP synthase. So hopefully this mechanism made a little bit of sense. Okay, and what we do by this enzyme is we generate reduced cytochrome C 2 times, which will then fuel cytochrome C oxidase, and that will also pump protons to fuel the proton gradient. Okay, so hopefully that made a little bit of sense there. What I want to do now is think a little bit about this enzyme in a little more analytical way. We have these drugs right here. One of them is called antimycin A, which is the top one. The other one, which is in the bottom, is mixothiazole. Okay, and in terms of this enzyme, there are different components of it, as we've seen. For example, we looked at the heme high potential, which you can also equivalently write H sub H, heme high potential, which ultimately transfers its electrons to oxidized ubiquinone. It's right here. Okay. So this right here, this is heme high potential, and it transfers its electrons into completely oxidized ubiquinone. So coming back here and looking at this drug, okay, antimycin A is able to bind to a site on the enzyme that effectively blocks the transfer of electrons from heme high potential to oxidized coenzyme Q, or ubiquinone. And so as a result of that, you can't generate the semiquinone intermediate. So this process is blocked. Okay, but it turns out that the heme high potential, so let's abbreviate this, H sub H in the reduced state, when it actually has the electron, it can actually transfer its electron, but to something else. And this is a really important point that I want to get across to you, is that these enzymes are not perfect. Okay, these enzymes in the respiratory chain, they can bind molecular oxygen because it's really small, and they can accidentally transfer electrons to it. So for example, if you have molecular oxygen, so you have molecular oxygen looks like this. So each oxygen atom has 2 lone pairs, and then each one has a radical electron on it. If you were to accidentally get molecular oxygen into the active site where the heme high potential is in the reduced state, you could theoretically transfer an electron onto molecular oxygen. And so what you'd end up getting is one of these atoms of oxygen now has a full octet. And this particular atom is this one, has a negative charge on it. And this molecule has a very special name. It's called, let me do this in red because it's bad, this is called superoxide. And superoxide is toxic. It's a free radical. It likes to react with things, and it'll destroy proteins, membrane lipids, and so forth. And so you actually have a whole enzyme to deal with this. It's called superoxide dismutase. We have a whole video on it. I think I'll put it next in the playlist just so you can kind of see it. But free radicals are bad. They're thought to cause things like aging, and so you need ways to get rid of them, and superoxide dismutase is certainly one way you do that. But this is a really subtle point, is that free radicals can be generated by the respiratory chain. And of course, this is a really bizarre case in which you'd be giving an organism antimycin A, which of course blocks the transfer of electrons between heme high potential and coenzyme Q. But this kind of stuff does happen on a regular basis, and you do have ways to deal with it. Okay? The other drug I wanted to talk about was called mixothiazole. Mixothiazole actually inhibits the transfer of electrons from reduced ubiquinol. So this molecule right here, this is coenzyme Q in the reduced state. It inhibits the electron transfer between that and the Riesk iron sulfur center. Okay? As a result of that, you again can't complete this mechanism. So this one also, for the most part, is a lethal drug that you would certainly not give to a human. Okay? So hopefully this gave you a little bit of intuitive sense on this enzyme, and hopefully you have a little bit more intuitive sense of the Q cycle at this point. Okay, so in the next video we'll look at how superoxide dismutase can handle free radicals, and in another video we'll look at also to see what other kinds of free radicals we can form and the harmful effects that they have. See you in the next video.